Hi folks, welcome to part 5 of the muscular system, the process of muscle contraction. The first 4 mini lectures led us to this point where now that we understand a little bit about the structure of skeletal muscle in the neuromuscular junction, we can think about the 3-part process of muscle contraction, which involves activation first. And as you can imagine, that's going to involve the neuromuscular junction. Next, excitation contraction coupling. So we have to hook together the action potential in the muscle, which is an electrical event, to the mechanical event, which is contraction of the sarcomere. And then the cross-bridge cycle, which is in fact what allows for the shortening of the sarcomere. So here's the Cliff Notes view. Activation involves the muscle fiber being stimulated by a motor neuron. Excitation contraction coupling involves the release of calcium from the sarcoplasmic reticulum inside the muscle cell. And then the cross-bridge cycle involves the formation of temporary bonds between the myosin heads of the thick filament and the actin of the thin filaments. So let's look at each of those processes in detail. All right, activation. So the first thing that happens is we have an action potential that comes flying down the terminal branches of this motor neuron. That would be A1. That's going to lead to the opening of voltage-gated calcium channels, right? Just like in a neuron. Calcium floods into the synaptic or axon terminal. That leads to exocytosis of the neurotransmitter, which remember is going to be acetylcholine. The acetylcholine diffuses across the synapse and binds to the acetylcholine receptors that are located within the motor end plate of the muscle fiber. And that is going to then eventually lead to the production of an action potential inside the muscle. Because remember, these neurotransmitter receptors are also gated sodium ion channels. All right, I'm going to play a quick video for you guys. And this video has a lot more steps in it, but for our purposes, we're just going, you already know steps 1 through 6 in this video because they're exactly the same as they are within the nervous system, right? So keep that in mind. Don't get, right, you might need to watch this more than once, but don't get caught up in all the details when you're first looking at it.
Nerve impulses, also known as action potentials, travel from the brain or spine to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so onward to step 2, which I've also called V here. So excitation, contraction, coupling. What we're looking at here is a lengthwise section through part of a muscle fiber. So we've got the sarcolemma, we've got thin and thick filaments, right? We also have the sarcoplasmic reticulum, right? Which includes these flattened endings, which are called terminal cisternae, and the transverse tubule system here. Remember that when you have a T-tubule invagination, which is an extension of the sarcolemma into the muscle cell and two terminal cisternae, then you have a triad. So the first thing that happens is that the T-tubules, right, which remember wrap each myofibril, conducts, because the inside of the T-tubule it's hollow, right? So what the fluid that's inside there is the same as the fluid outside the cell. So the action potential, which involves the exchange of ions with extracellular fluid, can continue, right? The actual excitation, contraction, coupling happens when you have the release of calcium from the sarcoplasmic reticulum where it's stored in response to the muscle action potential. So let's take a little bit closer look here. So you can see in this upper panel, we've got sarcoplasmic reticulum, the sort of blue-purple, the T-tubule, right? And so this would be the membrane of the T-tubule, and this would be the intersection. And these two are bolted together by this protein complex, which is voltage sensitive. it turns out that when you have an action potential come down the T tubule, that leads to this protein complex changing shape. That change in shape allows for the opening of calcium channels, which are another part. So the calcium here is, which is sort of hot pink color, is inside the sarcoplasmic reticulum, and you can see here it's sort of flooding out. So you end up with calcium flooding the sarcoplasm. And what that means is that calcium is actually the bit of matter that couples excitation, meaning muscle action potential, with contraction, with the shortening of sarcomeres. So let's watch that in a little bit more detail.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, now we're on to the third part of the molecular mechanism of muscle contraction, called the cross-bridge cycle. And this is a set of 4 coordinated steps. I'm not going to read through this because I'm going to show you guys a bunch of pictures, but for folks that like to see things written out, you can look at the PowerPoint notes that you have access to. One thing I do want to point out is that this is a cycle. Cycles by definition repeat, right? And so what that means is different sources you look at may start the cycle at different places, right? I'm going to assume that we've got ATP present and on the myosin head. Remember, the myosin head can bind ATP. So the first thing that happens is that the calcium, which we just talked about, was released from the sarcoplasmic reticulum in response to the muscle's action potential, binds to the troponin complex, all right? So this little binding spot on the troponin. And remember that the troponin sits on top of the tropomyosin, which is that sort of strand-like molecule. proteins change shape when things bind to them. All right, when calcium binds to the troponin, it essentially flexes, and that flexing leads to it pulling the tropomyosin away from the active sites on the actin molecule. And that's what this little GIF is showing us over here. The next thing that happens is formation of a temporary bond between actin and myosin, right? So the calcium has to be present in order for these binding sites to be able to come into contact. So a cross bridge is that temporary binding, right? For the cross bridge to form, the myosin head has had to hydrolyze ATP to ADP and inorganic phosphate, although they both stay attached at this point. Next comes what's referred to as the power stroke. So the inorganic phosphate is released, and that causes one of the hinges in the myosin to pivot. Then the ADP is released, and that causes an additional pivoting. And the pivot, because the pivot of the myosin head, because it's now attached to the actin of the thin filament, and because it's pivoting, the head's pivoting toward the center of the sarcomere, pulls the thin filament toward the center of the sarcomere. So that's where the sliding comes from. It's not really sliding, I suppose, because that implies things are slippery, and this actually requires a fair amount of energy. Now, I'm going to go back here for a second. If no more ATP is available and no ATP binds to the myosin head, then that cross bridge is going to stay intact. That bond is going to stay there. However, if you have another ATP bind, that allows, remember, proteins, when things bind to them, they change shape, or they can change shape. In this case, the binding of ATP leads to the myosin head changing shape slightly. This bond between actin and myosin is broken, and then this cycle can repeat again. So here's the same thing, but on a single page, right? We've got the release of calcium, which binds to the troponin, which moves the tropomyosin off of the dark green, at least shown here, the dark green binding sites on the actin. The myosin head has already been cocked because the ATP has been hydrolyzed, and we have formation of the cross bridges between myosin and actin, followed by the power stroke, which pulls toward the center of the sarcomere, and then finally the ATP binding. All right, so here we have, again, the cross-bridge cycle, a little snarkier format. Remember I said that it's a cycle, so you can describe it as starting at any point. One of the things we want you guys to be able to focus on, though, is the role of individual parts or individual molecules in this cycle. So what's the role of ATP? What's the role of calcium? What's the role of the myosin head? Okay, so take a look at this. It's the graphic is in slightly different order, but I think maybe because it's slightly simpler and because it's kind of like a cartoon, it's a little more accessible. All right, so now let's watch the cross bridge in action.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
All right. So hopefully with my explaining it, the explanation, and particularly the animation in the videos, you're getting a handle on this. One of the best ways to learn this process is to watch these 3 videos that are embedded in this presentation repeatedly. Watch it with the sound on, then watch it with the sound off, and describe what's happening. Right? Eventually, if you do that a couple times, you'll get to the point where you can sort of play the movie in your head, and then you can use the movie you're playing in your head while you're taking quizzes and exams. All right, last but not least, we're going to focus in on the function of the myosin heads, function of ATP, of calcium ions, and then we're going to come back out to look at the big picture, talk about what a muscle twitch is, and finish with whole muscle contraction.